For the next presenting company is Stem Cells, Inc., focused on harnessing and delivering the power of human neural stem cells into a wide variety of indications, including spinal cord injury and other CNS disorders. My theme is when you think about Stem Cells, Inc., or you think about Stem Cells, Inc., think about Police, that old song, a message in the bottle. In this case, stem cells in a bottle. And here to tell us about the stem cells in a bottle is CEO Martin McGlynn.
Thanks, Ren. So there's a device here. Here we go. Forward-looking statements, refer you to our Ks and Qs. I will be making some forward-looking statements. So I just quickly want to share with you what I'm going to try and cover today in a short period of time available to us. A little overview of the company, talk about our human neural stem cell platform and where we are in the development status, give you a financial summary, and ask for your order. Stem Cells, Inc. Has been around NASDAQ for quite a while, initially since 1992 as Cytotherapeutics, a company that was developing cell encapsulation technology, which acquired a small privately owned stem cell biology company that was started up in California by Irv Weisman, Rusty Gage, and David Anderson. And in the year 2000, Cyto sold off its cell encapsulation technology assets, and on the heels of a discovery in California of the human neural stem cell, changed the name of the company to Stem Cells, Inc. So we have been on NASDAQ as STEM ever since. We're headquartered in Newark, in California, 50 hardworking employees, and we are pioneers in the discovery and the development of an expandable, and therefore the cell in the bottle, purified population of human neural stem cells, which we call HU-CNS-SC. This cell is a remarkable cell. It has, of course, features, and I want to talk about the benefits because Jeff Jonas said we weren't to talk about science anymore. So this is a purified population of cells. We believe strongly in the motto that the purity of the population of cells that you transplant is really where you start to separate and make a difference in terms of observing the effect of a purified cell as opposed to any other extraneous cells or material. It's highly expandable. These cells grow like weeds ex vivo, and they're bankable, and we hold them in cryopreserved banks. In vivo, they self-renew. They are non-tumorigenic. These are an allogeneic approach, and it's a homologous use of cells. That is to say, we take brain tissue and we put the tissue back into the brain. And we also have a liver program, which I won't talk about today, where we take liver cells and we put liver cells back into the liver, so hence the homologous use. This, in turn, gives us a profile with the cells that essentially they're hardwired to become the specific cells of, or the building blocks, if you will, of the central nervous system, neurons, astrocytes, and oligodendrocytes. In vivo, they are lineage restricted, as I said. They survive, they migrate, they're regulated by the host, and they differentiate in a site-specific fashion. So you will only see neurons in a neuron zip code, etc. There are multiple mechanisms of action, if I might be so bold as to use that phrase as opposed to mode of action, because nature continues to fascinate us. But what we have seen in our work today is the cells are operating on a cell replacement with proper types of cells, such as oligodendrocytes, which perhaps you heard about this morning, neurons and astrocytes that are derived in vivo. And also we see a neuroprotection effect of host cells, and as well as integration into the host, we also see trophic effects. It's not too early to start talking about your label, as we also heard this morning. So when we think of the human neural stem cell, we think of a neuroactive cell-based therapeutic for restoration and preservation of neurological function in a broad range of CNS disorders. Our manufacturing process begins with prospectively isolating the cells. We use FACS sorting, and then we culture the cells, we grow them up, we put them into cell banks, and then we derive a final product. From a single master cell bank, we can derive multiple working cell banks, which in turn we delve into to extract the individual patient doses, and we can do hundreds of patient doses, even at the pilot scale that we're operating today. The cells that we are using in our clinical trial program are already in the banks, and so that's a significant de-risking approach. And the process is highly scalable for commercial use. So just very quickly on some of the remarkable properties of these cells in the animal world. And you heard a little bit about it today. In spinal cord injury, we see locomotor improvement that is dependent upon the presence of the cells, not only for the restoration of lost function, but also for the maintenance of that function that has been regained. Moreover, these cells have been demonstrated to be efficacious in the subacute setting, and you heard Aileen Anderson talk about the importance of that particular element earlier today. We have also evaluated the cells in various animal models of lysosomal storage disease, and we see the cells are capable of producing the missing enzyme cross-correction to the host neurons and preserving the host neurons. In myelination disorders, we've seen the cells develop into oligodendrocytes. They glom onto the denuded nerve axons and start to spin layer upon layer of functional myelin. And then in retinal degeneration, these same cells are able to preserve vision in a standard model, the Royal College of Surgeons rat, which is not a model for AMD per se, but it is used as a model for vision loss. And essentially, when you transplant these cells, subretina in these animals, they're able to preserve the visual acuity of the animals pretty close to normal. Pretty amazing stuff. And then last but not least, our latest program is in Alzheimer's disease. And here we've seen in two Alzheimer's disease relevant models the ability of these cells to preserve neurons and also to restore lost memory. This is obviously a very exciting observation. So that's the animal world, and this is all with the same cell, albeit at different doses into different parts of the CNS. We have three major areas of focus. We are pursuing and examining the potential application for the cells in the three elements of the central nervous system, the brain, the spinal cord, and the eye. We have completed 2 clinical trials, phase 1/2 trials, in very different diffuse CNS disorders. As I said, one in NCL, otherwise known as Batten's disease, and Pelizaeus-Merzbacher's disease, a myelination disorder, also known as a leukodystrophy, which we refer to as PMD, and the Alzheimer's program also, which I mentioned, which is at the preclinical stage, and for which we recently received California Institute of Regenerative Medicine award for close to $20 million to help fund an IND in Alzheimer's within 4 years. Very, very exciting stuff. And in spinal cord injury, our goal there is to have an application for these cells in traumatic spinal cord injury. We're currently working in the thoracic region. We've seen some incredible observations already, even be it from small numbers, and I'll talk about that in a moment. And of course, we're also, everybody's focus and goal is to move up the spinal cord into the cervical spinal cord region, because that's where the majority of these injuries are. And last but not least, we have a program, a clinical trial underway in dry age-related macular degeneration. And here, the objective of the exercise is to see if we can replicate in folks who have the dry form of AMD, if in fact we can preserve their visual acuity. Clinical observations to date. So to date, we have dosed about 16 patients, and we've put up to almost essentially 1 billion cells, 1 billion cells into the brain, less numbers of cells into the spinal cord and the eye. There's about 100 billion neurons in the brain, so this gives you a sense of the scale, and nobody else has ever done this, has put this many neural stem cells into a human brain. There have been no adverse events reported that are related to the cells, to the immunosuppression regimen, or to the actual procedure of getting the cells into the target site. We have data out over 5 years now with the Batten's patients. We will be shortly publishing data, 2-year data for the PMD The one-year data was very, very exciting, and we've demonstrated that all of the 4 subjects presented with de novo myelin and also with small but measurable neurological gains, which is quite unexpected in a disease of this nature, which is fatal and very aggressive. In spinal cord injury, in the thoracic spinal cord trial underway in Switzerland, we've observed multi-segmental gains in sensory function in the worst of the worst spinal cord injury patients. These are patients who have no sensation whatsoever below the site of the injury. We have also observed de novo myelin of the nerve axons, which I've already mentioned, and gains in neurological function. And all of this has been published in Science Translational Medicine, as well as the work that led up to support the IND with the FDA and the initiation of the trial in that myelination disorder. So next up, in PMD, as I mentioned, we'll report out the 2-year long-term follow-up data. We're hoping to see is the continuation of the gains in neurological function beyond the 12-month point up to 24 months. And we'll continue to follow these kids for another 3 years beyond this. We have on the docket a meeting to meet with the FDA so we can finalize a phase 2 study in PMD. And this, of course, would be a great landmark and a great accomplishment for the company. In spinal cord injury, we'll be looking to complete dosing in the study underway in Switzerland, and we'll continue to report out on an incremental basis data as we receive it. The nice thing about these is, yeah, there's no controls, but you don't pay a penalty when you report out and look at the data in an incremental basis. In age-related macular degeneration, our goal here is to complete dosing in the first cohort of 8 patients out of the 16. The first cohort are the more severely impacted. They're, for all intents and purposes, legally blind. So we don't expect that we're going to see many efficacy signals there, but the main objective of this exercise right now is the safety component. As we move into the second cohort, however, as we get to a higher level of visual acuity, and Gary mentioned, this is the paradigm and this is the way you have to work from a safety point of view, we would hope that we'll also start to pick up some signs of beneficial effect and biological activity. And in Alzheimer's, actually, we've already initiated the work, a lot of heavy lifting that goes into preparing everything that's required under GLP to submit an IND with the FDA. And this, once approved, would be the first ever human neural stem cell transplant in Alzheimer's patients. Again, the goal here is to transplant the cells into hippocampus. We're looking at memory function. We're looking at memory enhancement. The cells in the animals were able to go in and do the work that they did and restore lost function without having to reduce the burden, the plaque, or the tau. So apparently these proteins are not toxic to the neural stem cells when transplanted, which is important given the fact that diagnosis is early diagnosis in Alzheimer's is problematic. And by the time you are presented with symptoms, very often the patient has the pathology in the brain and has the burden already in place. So the good news is that these cells went into that kind of a hostile environment and nonetheless were able to exhibit their biological activity, and that, of course, is very exciting. Quick roundup on the financials. Picture's worth, I guess, 1,000 words. So we strengthened our balance sheet last year, and our pro forma cash coming into the year was $34.4 million. Not quite up there with others. I'd like to borrow a few bucks from Ed. I don't know if I can find him later, but he seems to be rolling in the cash, which is great. It's great news for all of us. Our cash burn, small incremental reduction in 2012 versus 2011. The investment thesis, Stem Cells Inc. is the most advanced cell therapy company in the CNS space. We have invested over $250 million to date in developing this technology. It's what I call doing the heavy lifting and doing it by the numbers, crossing the T's, dotting the I's, no shortcuts. What's exciting about Stem Cells Inc. today is we've now reached the point where we're starting to pull in the lobster pots and we're starting to look for the catch. And we're looking at early clinical data that's confirming the preclinical results. And that's critical, that we're actually starting to see in the human patient populations similar results and similar biological effect as we saw in the animal models. We are 2 to 4 times undervalued versus our comps. You probably have never, ever met a CEO who thought his company was overvalued, so I don't want to disappoint you folks. And last but not least, 2 points I want to leave you with. There's a wonderful opportunity to ride the wave as STEM appreciates in value to reach its level of comps. And last but not least, important near-term milestones will support appreciation. I thank you for your time, and I don't know if we have time for questions, but I'd be happy to try and answer any that I can. Thank you.
So unfortunately,